This tutorial shows how to load data and generate uniform manifold approximation and projection plots, UMAPs, in the Visual Genomics Analysis Studio VGAS. Begin by opening the application and selecting RNAseq and then Analyze. Select File, Open, and then select a gene.csv and metadata.txt files together. Make sure the gene and the metadata files are open next to each other on the left-hand panel, and then double-click the RNA-seq plot viewer option on the right-hand side. This will open a window showing samples at the top and then selected samples at the bottom. Samples can be viewed on the basis of sample and metadata, or just metadata. Metadata is set by selecting from the drop-down menu and then clicking Set. Select the desired cells in the top panel and then select Plus. This will move these samples to the bottom panel. The number of samples selected will be displayed. To generate a UMAP, select the required UMAP options from Euclidean, Manhattan, Cosine, Pearson, and Pearson 2. Or alternatively, select all models by ticking this box, and then select Generate R plots. This may take a while to complete depending on the size of the data. Once the plots are generated, click Generate Plot Viewer. This will open an RNA-seq Plot View Options window. Once the plot view has been generated, the data can be saved as a VGAS plot view file, .vgaspv, by selecting File, Save as Plot View, and then naming the plot view in the pop-up window, This allows users to open the plot view without going through the first step of selecting samples. Alternatively, if a UMAP or t-SNE plot has been generated previously using an alternative software, the user can select Skip UMAP and then load in the data without creating a UMAP. Once the RNA-seq plot view options window has opened, XY coordinates saved in an Excel as sample ID X and Y can be loaded to create a UMAP. To do this, select File and then Load XYs. Users can also save the XY coordinates of UMAPs that they've generated using VGAS.